We've looked at the overall structure of and organization of the nervous system. We looked at neurons. We looked at synapses. Time to look at another incredibly important cell in the nervous system, the glial cell. Now, glial comes from a Greek root word meaning glue, because when they were first discovered, it looked like they were what glued neurons in place. Think of the fact that synapses only work if you're releasing neurotransmitters right over the receptors, or they certainly work better that way. And so glial cells look like they sort of glued the synapses in place so that they weren't moving all around. And they do that, but they do many other things as well, and we'll talk about them as we talk about the individual cells. So there are 6 basic kinds of glial cells. Astrocytes are found only in the central nervous system. Astro means star. So look at in the bottom illustration there. See how it's shaped kind of like a star, the astrocyte is? Yeah, they're star. They're the star cells. They're the stars. And then microglia, say it any way you want. These are phagocytes. They wander through the nervous system, cleaning up debris and dead cells and things like that. Just think about that when you're trying to go to sleep tonight. Remember that you have lots of microglia crawling around on little feet. They have little feet called pseudopods. And they're crawling around in your brain. Instead of counting sheep if you're having trouble going to sleep, count your microglia. Oligodendrocytes, also all 3 of these so far found only in the CNS. Oligodendrocytes make myelin sheaths only on neurons in the central nervous system. Oligo means a few. So instead of putting down just one myelin sheath, they put down a few. Ependymal cells are very important. They're found only in the CNS, and they make cerebrospinal fluid. And Schwann cells, found only in the peripheral nervous system, they make myelin sheaths in the PNS. And satellite cells are structural support and also regulate ions and things, also only in the peripheral nervous system. So, astrocytes provide structural support, and they also are useful in helping to create the blood-brain barrier, and they also regulate ions. Microglia, again, are phagocytes. They eat up dead stuff in your brain, because remember, cells are dying all the time. You lose something like 1,000 neurons a day. Somebody's got to clean up the mess. The microglia do that. Ependymal cells line the ventricles and the spinal canal. They produce CSF. Oligodendrocytes form the myelin sheaths around axons in the CNS, just like we said a moment ago. And then here you see the satellite cells. They surround neurons in ganglia, specifically, providing structural support, supplying nutrients, regulating ions and neurotransmitters. So the blood-brain barrier is an incredibly important topic, so let's make sure that we're going to come back to this again and again and again. This separates the blood from the ECF of the brain. Now, sometimes on exams or quizzes, people tell me that the blood-brain barrier keeps blood out of the brain. Don't be one of those people. Your brain needs blood. Your brain is the highest glucose user in your body, and it gets that glucose from the blood. So no, blood has to get into the brain. But the brain is very choosy about what it lets out of the blood and into the brain. I mean, think about it. Your brain is making all kinds of important decisions. You don't want just any old toxins and junk getting from the blood into the brain. So the blood-brain barrier is a regulator. It's like a bouncer, all right? It's standing there guarding what's coming from the blood into the brain. And if it's not something that's supposed to be in there, then it throws it out onto the street, okay? Formed in part by astrocytic end feet and by tight endothelial junctions in the capillaries of the brain. So normally capillaries throughout your body are leaky. They're supposed to be. They leak out nutrients, glucose and amino acids and ions and so on. So capillaries are supposed to be leaky. But in the brain, we got to be especially careful. So the junctions, the endothelial junctions, endothelium lines the inside of the capillary, the blood vessel. So in the brain, the junctions are very tight, all right? And so they don't allow nearly as much out as would happen in other parts of your body. Restricts substances in the blood from getting into the brain. Only certain things are going to be able to get in, all right? This ultimately determines what's going to be available in the cerebrospinal fluid, all right? The CSF is what then flows through the brain and supplies nutrients and so on, carries waste products away. So the blood-brain barrier determines what gets into the CSF. So cells like whole bacterial cells and large hydrophilic molecules like glucose and amino acids, they cannot get through the blood-brain barrier. Now, do you need glucose and amino acids? Does the brain need those? Yes, it certainly does. So they're going to have special transporters. Whereas small molecules like gases and alcohol and hydrophobic molecules like some hormones can get through the blood-brain barrier, right? Alcohol has like an easy pass. It goes right through. It's like, it goes right to the front of the line. Trying to get into O'Malley's. There used to be Nick, the bouncer, used to let me right to the front of the line. He was a great guy. So, small molecules and hydrophobic things can get through. Hydrophilic and large things can't get through. Again, that's why you have to have special transporters for glucose and amino acids. One of the reasons that some drugs will work in the brain and some drugs won't is drugs that are hydrophobic can get through the blood-brain barrier. Hydrophilic drugs often can't. So, for example, a lot of chemotherapeutic agents will not pass the blood-brain barrier. That's why brain tumors are typically not treated with chemotherapy. There are some new chemotherapeutic drugs that will get through, but in general, most chemotherapy drugs can't make it through the blood-brain barrier, and that's why they're not used for treating brain cancer. Okay, so myelin sheaths, they insulate axons and increase the speed of the action potential. We've mentioned that before. So they speed things up. They're very important. They hold in the electricity. It's the same reason that, electrical wires. You don't see just bare wire all over the place. The things you're plugging in your computer and all the other appliances in your house. They're not just bare wires. They have insulation. Insulation keeps you from losing electricity to other places, keeps it from shorting out, and that's kind of what myelin sheaths are for as well. They hold the electricity in and speed up the action potential. The gaps between the myelin sheaths are called the nodes of Norve. And don't forget, you have to know that. For the next exam, we're going to have an oral component. I'm going to require you to turn on your microphone, and you have to say node of Norve. Remember, you have to say it like a French person. Turn your nose up and act condescending and superior and arrogant to other human beings. Schwann cells form a single myelin sheath in the peripheral nervous system. So you can see there a Schwann cell on the diagram on the right side on the illustration. You can see a Schwann cell makes one single myelin sheath. Whereas oligodendrocytes, see more in the middle to the left, you can see a cell there making 3 myelin sheaths. That's an oligodendrocyte. Now this is kind of a goofy diagram because they're simultaneously showing the CNS and the PNS. No neuron would be present in both. This is a diagram just of convenience to try to show you a myelinated neuron. And on the left side of that dotted line, they're showing you what it would look like if it were in the CNS. On the right side, they're showing what it would look like in the PNS. But Schwann cells, again, are only in the PNS. Oligodendrocytes are only in the CNS. So that neuron is a fiction that doesn't really exist. Now, regeneration is a cool thing. So out in the peripheral nervous system, what happens is, if you cut through an axon of a neuron, you'll lose function, you'll lose sensation, obviously. But what can happen here is in the PNS, the myelin sheath forms a regeneration tube. The neurolemma, the outer membrane of the Schwann cell, it provides a little tube that makes a passageway for the severed axon to reconnect, okay? So that's what they're showing there in the diagram. They're showing on the left side that in the peripheral nervous system, even if you damage an axon, notice how the Schwann cells can promote and guide the regrowth, and it'll grow back again. So if you've ever had surgery or a serious injury, you may know this. Like I had surgery on my wrist to repair my broken wrist. They cut through some nerves, some small axons when they did that. And after it was over, they regrew. But it's kind of funny because for a while the area was numb, and then bit by bit sensation came back. That was as the Schwann cells were guiding the regrowth of the axons. In the central nervous system, it doesn't work that way. The oligodendrocytes do not provide that neurolemma, that regrowth tube. And so what happens is when you sever axons in the central nervous system, they don't grow back. That's why spinal nerve injury doesn't repair itself. No, I'm sorry, not spinal nerve, but spinal cord injury. So if you damage your spinal cord and axons are severed, they can't grow back because the oligodendrocytes can't provide that regrowth tube. That's why, generally speaking, damage to the spinal cord is permanent, whereas damage out in the periphery will often repair. So here you see overall review showing the various kinds of glial cells. 6 kinds. I want you to know them. I want you to know what they do. So look that over, go back through. All of these will show up again later. So here you go. Which type of glial cell myelinates axons in the CNS? Would that be ependymal, astrocyte, Schwann cell, oligodendrocyte, or microglia? Yes, indeed. That is the oligodendrocyte in the CNS. The Schwann cell is in the PNS.